I wish.
Let me just, because I'm not at home, as you can see, or in the office, I'm just going to make you all co-host in case that cool startup internet here is going to kick me out. Great. So this is working. Here we go. 9:30, perfectly on time this week. Thanks for being here. We have Neuroccino today with Anna presenting what looked like a great paper to us. She very recently moved from the Netherlands down to Bordeaux, where she's now doing her PhD, and we're very happy that she volunteered herself to present Neuroccino today. So just a brief reminder, Neuroccino works the way that anyone can volunteer to present a piece of exciting science, may that be a paper, a book chapter, any other piece of exciting science you come across and want to talk about, and let us know. You're welcome to bring, and we have a chat about it. Without any further ado, Anna, if you want to take it away, if you can share the link in the chat so I can put it on YouTube. Perfect.
There we go.
That woman knows what she's doing. And then if you could share the paper in full screen, please.
Of course. You can see the paper in full screen right now, right?
I can see it, but it's still not full screen for me, but I can see it if that works.
What else can you see? You can just see the paper, right?
I can see like the paper. Yeah, much
okay,
better. Great.
Okay, awesome.
Thank you.
All right. Thank you for the introduction, Steph. So I'm very excited to present this super fresh work, which was published just a few days ago. Initially, I had another paper prepared, but yeah, I stumbled across this one, and then I just switched because I got quite excited. So there are people joining. Okay, so the paper is a Nature methods paper, but don't worry, it's not going to be too deep into the methods. It's called NEUROMAPS, Structural and Functional Interpretation of Brain Maps by Marcello, Hansen, and colleagues. And I got really excited because, put in very general words, it's a new approach to integrate different heterogeneous brain data, as for example, functional and structural data with other data as like data from gene expressions and receptor density, for example. And it basically allows the field and every researcher who uses the toolbox to place their data in a broader neuroscientific context, which is really great because yeah, we can now add information to our findings. So to demonstrate it a little bit more on an example, so imagine we have a neuroimaging experiment that we conducted and we find a result, let's say a certain brain area, I know Brodmann 46 or so, shows a higher neural activation for a specific task. And this activation is higher in younger patients or younger participants than in older ones. And we might guess now that, like, just from the literature and from our previous knowledge, we must that it could be a task that is related to working memory or just an attention task. But at the same time, we could possibly also oversee or just not know about other correlates that might have driven that, the result that we obtained. So that's age difference that we found might be because of certain cell populations or genes or whatever. So we are now still unable to see how those results fit into a broader context. And even if we can explain a finding from like our specific field's perspective, it's quite impossible for a research group that is specialized to basically integrate these findings into a broader context. So that's where the work from Marcello and Hansen comes in, who basically created the tool to integrate neuroscience data into one into one thing that you can basically generate new hypotheses and also answer and add to the knowledge that's already existing. So they said that hold on, yeah. So Such a tool basically needed 3 specific capabilities, right? They are saying it here. It needed a method for generating high quality transformations across multiple coordinate systems, which makes sense because different data is obtained in different coordinate systems. Then a curated repository for brain maps in their native space, and a method for estimating map-to-map similarity that accounts for spatial autocorrelation. And these were basically the challenges that they faced, or technical problems. And yeah, they managed to overcome them by creating their NeuroMaps tool. So the core of their work, what they did was basically having those, creating those transformations, or allowing for transformations across multiple coordinate systems. Then, like with this base, and yeah, of integrating different brain data into one, one map, or transforming it at least, so it's it can be all compared. It was made possible that heterogeneous data sets from the past, I think they took the past decades, so like the past 10 years, were integrated, and so they basically sought out a lot of data sets from a lot of different methods, and place them into the one same coordinate system, which is the library over here. And these data sets could then be grouped into 4 different subcategories, which is, I have it on another on another paper, but I couldn't put graph into it. So it's architecture, which is, for example, cortical thickness, right? It's the first brain here. Then we have the subgroup of cellular library, which is, for example, gene expression for from the Allied Human Brain Atlas, and like others as well. Then we have the dynamics, which is here, which is data from MEG, or just cerebral blood flow and cerebral blood volume. And we have the function data. So that covers, for example, evolutionary expansion and developmental expansion. And placing all these different data sets in the same spatial framework allows them to statistically compare their spatial profiles and to ask novel research questions and bring up novel hypotheses. But there is the difficulty that spatial maps can't be compared using just simple correlations, which was the default previously. Because the topography of neural data is spatially autocorrelated, right? So that means basically the features that are closer together in space inherently are inherently likely to look more similar than features that are further apart. And since nearly all data, like having that in mind, that means that all neural data is somehow spatially autocorrelated, and that kind of guarantees then correlations that will be found also between the neural data maps. And Markel and Hansen and colleagues addressed this problem by applying several approaches for performing basically spatial permutations for significance testing in their tool in NeuroMaps, and that helped to minimize the inflated p-values, which were which would have been obtained by just using normal correlations. So basically that spatial autocorrelation problem was overcome by their method. Here you can see. Here you can see all the brain maps from the published literature, which they have in their toolbox so far, because they're also saying that it's just the beginning, obviously, and not the end. That's basically here. It's described like that they use the spatial, multiple spatial node model frameworks that made it possible to account for the autocorrelations. All right, and so how they basically demonstrated their that the toolbox works. It's basically also just a package in Python, and it's open source, which is also great. I didn't have the time to check it out yet, but I'm really excited to look into it a little bit more in detail and just play around with it. Just feel free, it's on their paper. Click on, there is a link somewhere. So how they, having the transformations and having overcome the autocorrelations problem, they now tested their toolbox, their approach, and demonstrated for what it can be used. So this is a super interesting graph here, which I'm gonna go and into more detail in a bit and explain to you everything because it's really great. So they ran some tests basically to check how this data integration of heterogeneous data would work. And a very cool example that demonstrates it beautifully is one that where they used basically an open source data set, like a just random data set from, not a random one, but a data set that was not, that didn't have didn't play any role in their paper or in their methods here. The dataset was contained of cortical thinning in patients with chronic schizophrenia. So that's what you can see here. And so we just take a look on the first row of the figure. And so they estimated the cortical thinning by applying deformation-based morphometry, and like they basically estimated the cortical thinning, right? And then they calculated the extent of gray matter expansions or contraction in patients relative to controls. And then they used NEUROMAPS transformations to convert the MNI 152 space, which the data was in, to the surface space of each of the they selected 13 brain maps from the NEUROMAPS, which are target maps. Like they selected 13 of these and transformed basically their map to the target maps to see how they are correlated with each other. And they were able to show that the spatial topography of cortical thinning is greatest in regions that show the greatest neurodevelopmental expansion, which was basically obtained from a completely different data set. So if you look at the graph, then you can see like the red dots represent significant results, right? And these are the 13 maps that they use. And you can see at allometric scaling and NIH and PNC that these are significant, right? And going back to our legend here, that's the brain expansion. So allometric scaling here. And the second, looking at figure B, the second row, it's another example where they proved that data integration works. So it's where they showed that the brain regions that have the greatest evolutionary expansion. So they looked at evolutionary expansion, which was obtained from macaques. So they integrated macaque and human data. They showed that the regions that have the greatest evolutionary expansion also have the greatest inter-individual variability in regional functional connectivity. So looking at here intersubject variability, it scores, as far as I remember correctly from the paper, it's obviously here above 0.5, but it's I think 0.58 even, the positive correlation. And so that's there is a huge correlation between the brain expansion and inter-individual variability. And then again, like completing the picture and looking at what that actually means is so they integrated basically then the literature as well, saying it's consistent with knowledge that the greatest inter-individual differences in brain structure are in phylogenetically more recent transmortal model cords. And another explanation, so like another thing that they where they integrated the literature really beautifully, I think, is that they found also a negative correlations with the first principal component of gene expression, which is here, right? It's a quite high negative correlation. And this is consistent with the idea that phylogenetically neocortex is characterized by divergence from genomic gradients. So so they could basically test their approach and then basically put that into a bit bigger picture. So yeah, that was what got me really excited about the whole approach and the application. And I think that bringing together different and heterogeneous data types in a unified framework is, as they also say, just the beginning, because it can just be, it could be like further fed with like more data and more maps. And this can lead to not only answering questions or research questions better and understanding things better, but also for generating new hypotheses. So that has been at least what is said, that it has been an obstacle for a long time to integrate this data, because bridging between like different scales of different neuroscience data has been quite difficult and not really possible on such a broad scale up until now. And I think it's a beautiful integration for neuroscientific data. But there's also what I liked in the paper is that they take also a critical view or a critical argument to their approach, saying that, of course, like integrating massive amounts of data is not a guarantee for mechanistic understanding, but it's also not really possible to obtain a system level mechanistic understanding without bigger amounts of data, right? So it's basically somewhere a balance has to be found. And that's why it's not not an end, but just the beginning. Another caveat that they're talking about is, of course, that with data-driven approaches, it's always a little bit tricky, because obtaining or feeding existing data into a model, or building your work on existing data might also lead to that data reflecting historical biases and historical mistakes in science or research trends in general. But here again, just the beginning, not the end, because with more data, these maps should improve, right? And they will allow then for like more fine-scaled links across sub-domains of neuroscience. And that's the future of NeuroMaps would be to include more maps, for example, from single neuron electrophysiological properties, or laminar profiles of like different cell types. And it's also a cool approach to like not only basically it's the data integration, but for example, it can be used for finding out whether there have been things that have been historically overlooked over the years. So for example, in different diseases, whether there is, for example, a cellular or like genetic driver to a certain disease that has been that just has not been found yet. So it's not only for testing already existing hypotheses, but also for generating new ones, which I think it's quite beautiful. And so that's the last figure, is just the application of the, I think, 20 maps they had. And the interesting thing is that they cluster in two different, you can see my cursor, right? Can you see my cursor? Okay, cool. They cluster in two different clusters, which is yeah, so basically the more anterior posterior cluster and the more unimodal transmodal cluster. And this kind of also suggests that these brain phenotypes reflect a fundamental organizational principle of our brain. So I think that's I think that's a super cool paper, and I'm super excited to hopefully at some point try it out and to maybe contribute a little bit of to getting neuroscience known for being data rich and theory poor, or at least yeah, that's what they say, to data rich and theory rich science. Yeah, I hope you're as excited as I am, and I hope I can answer your questions. So fire away.
Thanks, Anna. Amazing. Thank you very much. Are there questions in the room, comments, points of discussion, anything that sparked your interest and you think you could take it further? Wow. 2 hands are up. I don't know who was the first one. Chris.
All right, thank you for presenting this paper. It looks very interesting. I just have a question. So do you know Neurosynth?
Yeah.
This paper reminded me a lot of Neurosynth in kind of the purpose of it. So I guess it's a little bit more technical in because they transform data sets, and it's not just scraping articles and finding coordinates. It's a little bit more complicated than that. But I'm thinking, do you think it would be it could be used in combination with Neurosynth to have, because for now the paper looks a little bit limited in the amount of data that they have, and the questions that could be answered with the tool. But as you said, they want to expand it. But I'm thinking maybe a link with Neurosynth could kind of bridge the gap between low the low number of data sets and more. There
already is a link with Neurosynth as far as I understood. So they integrated the Neurosynth data. I am sorry for scrolling. They already integrated it somewhere in one of these maps. I think either the function map, like one of them is also driven by Neurosynth. Yeah, like, so the Neurosynth data is already integrated, and it's just expanding expanding the brain maps to more data, so we can feed or see more things.
So that makes sense to have a link.
No, absolutely, of course. But yeah, it's more a little bit, it covers just more brain maps and just more data.
Yeah.
Which is cool. Yeah, Michelle.
Well done. Yeah, Michelle has a question.
Yeah, I have a comment and a question. So the evolutionary expansion map, they're not really evolutionary because they're the expansion from a macaque to a human. The macaque brain is not, our ancestors' brain. Macaque have had like their own evolution going on in parallel of the human brain. So it's just, before people get too excited, this is not like, it's not exactly an evolutionary expansion. It's a comparative expansion map.
Is that your comment or your question?
That's the common part. My question is, like, is there software online that can you show it to us?
I wish I had time to check it out myself. I didn't. Unfortunately, it's not a software online. It's a package for Python. So it's an open source package.
Okay, I see.
So it's called NeuroMaps, and exactly. So they just it's a bunch of code. and script.
And I see that like so everything is converted into a surface mode. So
Yeah.
my question really would be, how about subcortical areas and white mapped pathways? Should we just give up?
I am not sure here. I think that's just for visualization purposes. But
Okay.
here, yeah, so that's basically how they transformed everything, also with different resolutions, like on different resolutions. But I think I mean, with structural, with like functional gradient, for example, they must have also something that's not just cortical, right? And also, if you look at that, okay, no, that's just central sulcus. But I think they have, they don't only have the cortical
Okay.
areas.
Cool. Thank you. It was a great presentation. Thank you.
Thanks. And with about the evolutionary extension, I think, or maybe you can help my my thought process. Would it be even more like, could it be an evolutionary expansion if we would add, for example, chimp data and, I don't know, baboon data into that? That would be then evolutionary expansion, no?
So so it all comes from the models that the analytical models that you have behind your comparative anatomy. So if you do a direct comparison between the species, you're doing comparative anatomy. But if you try to estimate the mid deformation between your different species, then you have an estimation of the common ancestor from which we can derive those expansion. But nobody's doing that really, except maybe Rogier.
Okay.
And what's his name in Paris? Forgot.
Yelek or something? Never mind.
Yeah.
Do you mean Jérôme Zalé?
No.
Yeah, I meant Jérôme.
No. On the morning, I forgot his name. It's all right.
It'll come to you. Whilst you ponder the name, we can go to
Roberto Toro. There you go.
Roberto. Okay, that was too obvious. Hervé also has a question.
Yes, thank you, Anna, for the presentation. My question is in continuity with the one from Michel. So I was looking at the standard coordinate file system, because you say at the beginning they used 4 different systems. And so my question is, did they compare the different approach looking at volume-based maps as compared to surface-based maps? So as Michel said, it's sometimes cortical surface map, and if you want to look at volume-based maps, it can be different. Do you know if they compare them, or they try to see if there is a difference when you have maps, voxel maps or surface maps?
I don't think they did a comparison. So basically the transformation part is about transforming maps of different spaces into one, or like at least trying to. So they have the MNI space there, right? Then the FS average, FSLR, and the Civet. I'm not sure whether I'm pronouncing it right. So as far as I know, the Civet is the surface, right? It's for surface, but MNI would be everything, no? So I'm not sure whether I can answer that question really good, but what they did is that they looked at correlation matrices for the 20 brain maps. So not only for the transformation brain maps, but for 20 brain maps in the toolbox for each surface-based coordinate systems. So it's I think, yeah, the focus is more surface-based. I'm not sure whether this answers your question right now, but
yes. Yeah, I know at the beginning of this work on templates and the standard space, there was a lot of work to trying to have a common space which represent the middle one. And for example, Peter Kuchunov was working on that and trying to get something. I think here it's more, so now we are more looking at in the literature, the people are more working on surface-based space. So it makes sense to work more on this kind of thing. So, and in fact, when you look at these different space, I think they are all based on the MNI space, because even if you look at, I'm not sure, but I should be look at that, but all of them should be registered to the MNI. There is still differences because some of them are diverge from the initial MNI space. But,
yeah, you mean the like FS average, FS LR? Yeah. They are, for example, also FS LR was initially also MNI, some like not MNI 152, right? But it was like MNI something. So it makes sense that they basically because I guess the transformations would be then more reliable or like easier.
Yeah.
Right. If it's like if it's already similar.
And remember, the first space was made by a French guy, Tarak, and so all of the even MNI was based on this Tarak, and so, but you know, Tarak was only one hemisphere, old woman, and so they started to look at something more representative of the average population. So now we are at the MNI space. Time.
Thanks
also,
for that reminder. Just a quick note, I'm going to keep
Should we just think together?
I have to run, but we're going to keep the discussion going. I leave you in the safe hands of Michel Thibault de Schotten. See you next week.
Okay,
see you. Bye-bye.
So she's going to nice the moment. 3, 4.
After the Nobel Prize, we can
Valentina, question.
So I was wondering, how do they measure the unimodal, transmodal organization, hierarchical organization within the maps and the anterior posterior?
That's a good question. I will not be able to answer.
It's okay. I was wondering whether you said it, or I'm gonna go and look for it in a paper.
I believe they say it somewhere, but it's really nothing they put the focus on. So I am not sure right now.
I'm sorry. Thank you.
Oh, good. Yeah, so there's, across all examined systems, we find that the brain maps tend to form distinct clusters that largely recapitulate, a relationship involving anterior, posterior, and unimodal, transmodal axis of variation. So it's, so one cluster, no, that's just the explanation of the clusters. It's basically on the first, the last page, but they don't say how they do it. So they, there is no explanation, unfortunately.
I'm sure it's there somewhere.
If you say so, maybe in the end with the methods that they
exactly. Probably yes.
Thank you. The nerds. All right.
Any additional questions?
Doesn't seem to be the case.
I had another comment. I think I remember a study from Croxon in 2017. Sorry, I'm dragging the cover a little bit, mentioning that inter-individual viability and evolution were related. So I found it good that it was confirmed in this paper. It was really very interesting. Other than that, I think it's a great initiative that try to integrate different fields and different maps together in order to try to get more than like the unimodal interpretation of data people can put their hands on. So that's good. It's like a shame it looks like it's missing white matter and subcortical structure, but as they say, they want to develop the software further. The integration of structural connectivity with surface representation And also the use of subcortical structure with surface representation is not easy because those are really three-dimensional volumes. You can't really flatten it up.
Mm-hmm. But imagine also having, for example, maps for certain clinical populations, and then say stroke patients or Alzheimer patients, or like they already integrated the schizophrenia thinning maps, thinning maps, and just imagine having that for stroke. How cool would that be to integrate basically all these all these different data for
And does it require, in terms of like making it run, does it require to install a lot of library or use a Docker? Or
I really wanted to check it out, but I found the paper on Friday and I had a different one prepared actually, and then I got so excited about this one that I just wanted wanted to check that out, but I didn't have the time to look into libraries and to download it and to play around with it.
It seems pretty simple to install. Just check the GitHub. It's just like standard libraries. NumPy, scikit-learn, SciPy, NiLearn, Nibabble, and Matplotlib. That's basically it.
You need CVET and FSL, no? Don't you?
They don't. I don't know the extent of the tool. I don't know if they actually provide the normalization tools.
Okay. Maybe they just apply the deformation.
Yeah.
No, that's a cool initiative. All right. Any additional questions? We good? Well, in that case, that's a wrap. Excellent presentation, Anna. Thank you very much. And we'll meet again next week for the Neuroccino at 9:30 a.m. What's awesome last week in science, just bring it on and get us excited for a fantastic week in science.
All right.